I'm Sergio. I'm coming from Mats Nilsson's lab. We work with in-situ sequencing data, and the name of the lecture is in-situ sequencing data analysis, but actually, yeah, many of the lectures that you're doing during these days are focused on that. So I'll be focusing on some aspects that haven't been that covered so far, which are cellular segmentation, cell type prediction, and gene imputation. And so starting with the first one, I'll just add a slide of what is the basic output of the coding of in-situ sequencing data and all the image-based spatial transcriptomic methods. And you know that, because we have several lectures on that, but basically what we get is a CSV containing the X, Y, and potentially Z positions of the spots, like here, and then the identity of every spot, plus some additional quality metrics sometimes. And then together with this, one typical thing that we do in the microscope is to image as well nuclear staining. Commonly, we use adaptive staining, and we will use that for cellular segmentation later on. So that basically what we get from the decoding is this type of drawing, this type of picture over here. So what is the meaning of segmentation in the context of the spatially resolved transcriptomic methods? So the overall idea is that we are going to use the adaptive staining that we have to delimit every individual cell that we have in our tissue. And then with that, we are going to assign the different reads that we have placed in the XY position to an individual cell. So this task can be extremely easy, or it can be extremely difficult. It depends mostly on the quality of your tissue, and also the structures that you have present in your tissue. And for this reason, there are several types of algorithms that can be used. And some of them, every algorithm has its own benefits and its own cons. So we're going to go basically through the different approaches that we typically use for these tasks, from the most easy ones you can do to the most difficult ones, or the most complex ones, but also the most interesting ones in a sense. So maybe the most trivial thing we can do with our data in this context is to, now it's frozen. Yes. The most trivial thing we can do with our data, which doesn't actually rely on the position of the cells, is to just do binning of the data. Binning consists of basically placing equally distributed bins through our image, and then considering that all the reads that fall within a square or a hexagon in our tissue are assigned to a specific spot. This would be like transforming our data in sort of like a ST-like data, where we have not only a single cell in a single bin, but several. This is, of course, not the most effective way of analyzing the data, because one of the benefits of in-situ sequencing and most imaging-based approaches is that you can actually resolve single cells. But it can still be useful in cases where nuclear segmentation is impossible, or in cases where the quality of your tissue and therefore the number of reads that you have detected are not that informative, so that nuclear segmentation would lead to have a lot of cells with a low number of assigned reads. But if we go to actual cell segmentation, maybe the most common and historically most used algorithm for nuclear segmentation is a watershed segmentation, and this is a method that's consisting on the trying to delimit every cell in the adaptive staining by looking at minimums and maximums in intensity within the image. So basically, I'm representing sort of what it would be a nuclear staining in a 2D over here. Sorry, in a one dimension. So in the x-axis, we have all the different pixels in one dimension, and in the y-axis, we have the intensities. So of course, in a nuclear staining, the regions that are stained will have peaks, and the regions that are not stained will have valleys. So they will have minimums. In this case, it's the opposite way. So we will have the minimums in with, I'm trying to put my, okay. Yes, so in here, the minimums represent the peaks in intensity and the maximums are the absence of intensity. So the idea is that we can try to assign cells to define cells by groups of pixels that would fall if we're considering a drop falling into these mountains and valleys. We would assign all the pixels to a single cell if they fall into the same sort of valley or minimum, a local minimum here. So this is the most basic way of assigning, of detecting nuclei. And then what we typically do on top of that, since we know that cells are not defined simply by nuclei, because we lose a lot of the cytoplasm in that, we typically expand the resulting masks to include also the reads that would be around the nuclei. The problem with this, again, is that we are not considering the cytoplasm correctly. We are just expanding the masks that we take from our nuclei. And another problem is that this can fail in very dense areas where we have a lot of nuclei. But it's a quite fast and easy to apply algorithm that can be very useful in cases where we have quite sparse cells and we don't have any areas where it's difficult to segment. When we have nuclei that are quite compact in the tissue, because of the structure that we are trying to image, there are several papers that came out with different deep learning based algorithms that have been very useful to actually detect properly the nuclei in especially the denser areas where we have a lot of nuclei. Within these algorithms, we can find cell pose, elastic, which is this one over here, or stardist. These three are quite popular. We'll be using in the lab. We'll be using cell pose for this task, for the segmentation. And the advantage of those is that we can actually train the model out, either use a pre-trained model that has learned what a cell means and has learned from previous cases how to detect. It's actually good. It's a model that is good in trying to detect nuclei. And then we can apply it to our data because it will usually perform good. In these models, one of the things we can also do is to train it ourselves. So I'm going to show a small video. Can you see my screen? I guess yes. So some of these algorithms, like elastic or cell pose, allow you to actually draw on top of your images what is a nuke, what is a cell, and this can be very useful in case the model that you have pre-trained is not useful for your case because of the characteristics of your image. So they are very versatile, and they are becoming the gold standard in cell segmentation for spatial transcriptomic data, but also for other types of data sets. So the problem with this, so the benefits and the benefits of this algorithm is that the nuclei detection is way better than the watershed segmentation, especially in denser areas. But the cons is that still we are not considering the proper shape and location of the cytoplasm of the cell. We are only relying on the location of the nuclei. So really recently, there have been several methods that are relying, since we actually have the location of the reads, they are relying on the location of the reads to basically establish the shape and to delimit the cells. And you had a lecture yesterday on this method developed in Germany, this SSAM. That is one of the approaches that doesn't rely on it's a segmentation-free algorithm that can allow you to detect the different cells and the different patterns. But there are many others that are quite useful. This figure is extracted from the Bezier manuscript, and Bezier is a method that actually relies mostly on the gene density in your tissue but also to in the different cellular composition to segment individual cells. So you can see in this panel C, you can see, for example, in the x-axis the distance to the 16th nearest neighbor that every single read has, and the density over here, and you can clearly see that the peak, the first peak represents reads that are intracellular, and then we have the second peak in green that represents background, and we can actually make basically see where both curves the intermediate peak between both basically, and we can use that to establish the shape of the cells, and therefore assign reads in a much better way than simply using nuclear staining. The idea is that with these methods, of course, the benefit is that we will be much better in capturing reads that are outside of the nuclei, which is in most of the cases is a wide proportion. The inconvenience of this is that in very dense areas where we have a lot of cells that have the same cell type, or they have a similar cellular composition, it will this type of methods will usually fail, because it will not be able to distinguish one cell from another, especially if both cells are very similar. In the case of Basor, for example, we can also include a cellular segmentation based on DAPI that will complement the simply read-based segmentation, and we can also give it a specific importance in the segmentation task. So I think that's where most of the field is moving through. But as a summary of all the strategies, as I said at the beginning, all of them are really useful, and then can be really useful in different situations. So it's quite important to understand what is the nature of your data set, if it's dense enough, if you have a tissue that have structures that are difficult to resolve. And then based on that, you can decide what's the best strategy to use for cellular segmentation. Yes, so after doing cellular segmentation, we will end up in a sort of output that looks like a cell by gene. It's actually a cell by gene matrix where we have all the individual cells in the rows, and then every column represents every single gene that we have, and then we have an additional columns for the coordinates, X and Y position, and then depending on the method that you're using, you can also have information relative to your specific cell size or cell shape or all this type of metrics. Then the typical thing we can do with our cell by gene matrix is to convert it to a single cell sort of object, and we can do this analysis with different packages. In here, you've used Undata, so you've used ScanPy because we're working with Python-based tools, but you can also do this with Seurat or other R tools. And then you can basically treat this data set in the same way you would treat a single cell data set, with some sort of modifications I will shortly mention. So the first thing we need to do in our processing steps is to filter out cells based on the counts. We can have, especially if we segment based on DAPI staining, we can have cells that do not have any read assigned, and we need to filter them out because they are going to bias our analysis. We also want to exclude cells that have too many counts, too many reads assigned to them. Those could be caused by missegmentation that would lead to doublets caused by this missegmentation task, and this can happen in places where we would under-segment and we would put several cells together, and therefore this usually the cells that have too many counts in this type of data sets are artifacts, and we need to exclude them. And then we also want to, for clustering purposes, we also want to exclude cells that have too few genes on them. So after filtering this information, what we are going to do is in a similar way as we did in the single cell RNA-seq, we would log transform our data and normalize the counts, following with a scaling of the expression. Despite we have seen empirically, that the scaling of the data sometimes, depending on the type of data set you have, it doesn't help much, and sometimes it even increases the effect of sparser genes or in the clustering. So we are. I'll just put it in gray because we are doubting a bit about the utility of this step with our type of data, but that is still hasn't been analyzed properly. And after these transformations, what we can do is clustering in the same way that you would do it with a single cell data set and with your favorite algorithm. And basically the idea is that after you perform clustering, here I'm representing my clusters in a UMAP in dimensions. So you can see every cluster is a different color. The difference with single cell is that, of course, we can translate these clusters into the space. So we both have at the same time, if we do this this workflow, we have the low-dimensional representation of our data and also the actual representation in the tissue, and that can be very, very useful for several types of analysis later on. This type of task is, of course, not relying on any prior, but one thing that can be very helpful when you are analyzing a single spatial data set is actually to have a reference single cell or single nuclei RNA-seq. And there have been a set of techniques developed that which aim to integrate single cell RNA-seq datasets with spatial transcriptomic datasets like ISS, or even this set of tools also work with ST-Visium. So one of these techniques is Tangram, and that's what we'll be using in the lab today. The idea of this is that we will basically combine our single cell RNA-seq and our spatial data set using the set of common markers present in both data sets. So in single cell RNA-seq, we'll have a close to whole genome, whole transcriptome sequencing. So we'll have all the genes. And in the case of ISS or other image-based techniques, we will have a much more reduced panel. So that would be the limiting factor. And we will use the common genes in both panels to integrate them in a common embedding space, and therefore we'll be able to relate cells from single cell RNA-seq to cells in the spatial analysis. And this can be very useful because with this type of tools we can both assign a specific cell type and look for cell type patterns within our tissue. So we'll basically going from the single cell RNA-seq to the spatial data. And another interesting thing we can do is to actually impute the gene expression from single cell RNA-seq into the space, meaning that since we have a limited panel of genes in the spatial data, and we have all the different expression of the genes in the single cell RNA-seq dataset, we can take the genes from single cell RNA-seq and try to translate those genes into the space using the common embedding we've made with this type of tools. And this is what you can see actually here. First, this comes from I think the Tangram paper as well. You can actually see here how they've used this method to basically place the different cell types detected in single cell RNA-seq into the tissue. And here you can see the different glutamatergic cell types within a mouse cortex, I believe. In this case, using Murphy's data set. And that's one of the tasks you can do. But the other thing you can do is to actually impute the expression of new genes that were not in the original panel. And you can see here the comparison of the actual detection using Murphy's and the the prediction that the Tangram in this case has given. So we can see that the patterns for most of them are actually very similar, and that we are actually good in translating or imputing these gene expression patterns. However, in this, I'm gonna go, I think, here first. Yes. These methods, these integration methods, that rely on the integration of single-cell RNA-seq and especially resolved transcriptomic datasets are very nice, but they have some priors. First one is that they require that your single-cell RNA-seq dataset and your spatial dataset are comparable, both in terms of cell types present, meaning that, of course, if I don't have a single cell cell type present in my single-cell RNA-seq dataset, I will not be able to detect expression of that pattern in the space. But also some of these methods, and not all of them, they rely on also cell type proportions. So they are assuming that the cell type proportions that you have in the single cell dataset are gonna be equivalent in the space. And another thing you need to take into account is that your integration will be dependent on the quality of your data. This means that an incomplete panel that doesn't, now I'm talking about spatial, an incomplete panel that doesn't include all the proper markers for the different cell types that you have in your tissue might bias the integration or lead to a bad integration. And also a too sparse data set where you have a lot of genes that haven't worked properly or that are not detected specifically enough in your spatial data set might also lead to inclusive results. And this is very important to take into account because they can be very useful tools, but if they are used wrongly, they can lead you to results that you cannot actually trust. Yes, and I'm gonna also talk about another, now after going through the different segmentation approach and also cell type prediction approaches and gene imputation approaches, I'm gonna also talk about an approach that sort of combines both of them, and that's called probabilistic cell typing. In this case, what we have is nuclear staining, and we have used the nuclear staining to segment the cells. And basically this model, based on single-cell RNA sequencing data, we will extract the gene the expression signatures for all the different cell types that we have for the genes that we have in our spatial experiment. So basically, I will extract the expression patterns of the different cell types I have in my single-cell RNA-seq experiment, and I will try to assign in a probabilistic manner a cell type to every cell in my in situ data set. The idea of this is that I can do these tasks of both segmentation and cell type prediction in a single step, so that I can basically first assign the cell types in a manner that doesn't rely on the cell type proportions of your cells in the single cell RNA-seq dataset. And second, it will allow us to some extent assign reads that are outside the cells to cells that they belong based on the cellular identity of that cell and knowing the priors that single cell information gives. And regarding that, I'm gonna also show you this. How does the output of this look like? And this is another viewer. We can basically see the small spots are the reads that we have detected, and then in the different colors in the segmentation masks, we'll have the different identities of every cell. And basically, if we click to a specific cell, for example, this one, we'll be able to see that all these reads have been assigned to this cell, and at the same time, this cell has been assigned to this very specific cell type with a certain confidence, because this is a probabilistic method. So we actually get the probabilities of every cell type, of a cell belonging to every cell type. Yes, so yeah, going back Yeah, I think that was it. So I'll be happy to take questions on this. Otherwise, I think the next step would be to go and try it yourself in the lab. So thank you.
Sergio, thank you. Can I ask a question about you mentioned already, and I think we talked about it yesterday a little bit as well, and that the choice of assay also biases the type of data you're getting. So you hinted to it already. So if it's incomplete, right, or it's a small data set, the integration might fail sometimes. In your experience, have you found any differences between a single cell and single nuclei data set? Because the nuclei might be limited, at the same time the segmentation, it might help actually define it to a region, which is the nuclei, which you have in the DAPI. So what is your experience and the integration? What is best or things to look out for?
Yes, of course, the better the original data set is, the better representation of all the cell types that you have, the better the integration will work in that sense. So we have, from our experience, we have seen in single nuclei RNA-seq some cell types that were not present in single cell RNA-seq. So in that sense, single nuclei would work better for the integration in just in the sense that we will have all the cell types represented. Still, you need to think that in the space you are actually going to detect all the cell types that are present there. So we are still not sure, and we have found in some cases some differences even when using a single nuclei RNA-seq. Some cells that are actually present in the tissue, but not present when dissociating it. That's quite common, and I think there are not any methods still that can help you to actually know if your single cell RNA-seq data set is representative of your spatial data set or the other way around. So it's still work that's going on. There are some papers published recently, but I don't think there is a clear answer so far.
See, it is clear from your, it really depends on the quality of the data set. There's not single nuclei or single cell. It's really the complementary of it once more, indeed, which we need to look out for. And the loss of cell types, I think that's the bias then to be mindful of. That's clear. Yeah.
And also the distributions of the cell types. That's also one thing that is very important. So the ideal case for these methods that rely on integration would be to have one sort of chunk of tissue, piece of tissue for single cell, and then just the sections next to it for spatial. That would be the ideal case, because then you are mostly certain that the cell types distributions are going to be similar, but still you are not taking into account the dissociation bias.
Perfect. Yeah, thank you.
All right. Thank you so much, Sergio, for presenting this today and helping us also with the course. Now I think we can then go to the exercises. In the schedule, I added also a link to download the single cell data if you didn't manage to run it yesterday, or if you have any problems. But I just need to warn you that it's 1 gigabyte, so it might take a while. So, but then, yeah, if you had any problems in the previous exercise for analysis of the single cell, you can download it there directly. But in any case, I think you can also already start with the ISS pre-processing while that one is downloading, because you need that later on, if I'm not wrong, right, Sergio? Oh, sorry, you're muted? No? Yeah.
What